Hello everyone, my name is Khushbu Gandhi, working as a technical officer in microbiology department at Elizabeth MacArthur Agricultural Institute. Thank you so much, Belinda. So far we have all come to know about the bee disease diagnostic world at EMAI. Let's move to the second phase, which is bee research at Elizabeth MacArthur Agricultural Institute. Today, I'll be talk about the three different research topics that has been carried out in last 3 years at EMAI. Very first one, has Penibacillus larvae, the curse of American foulbrood, had developed the resistance to the oxytetracycline in New South Wales? My second topic is about the introduction of the newly developed test at EMAI which can detect OTC in powders collected from AFB treated beehives. And my third and the last, very important, to develop a technique to trace the bacterial disease of honeybees by using their genomic sequences to control AFB infections and outbreaks. Let's begin with my first topic. Now, we are all aware of the use of the antibiotics for the treatment of AFB. In 1940s, the sulfonamide were the first antibiotic used for the treatment of AFB, but Penibacillus larvae developed resistance in the USA. In 1950s, the use of the tetracycline commenced. However, Penibacillus larvae has also developed the resistance in some countries, including Argentina, Canada, and USA. As we are all aware of the use of the oxytetracycline is illegal in mainland Australia for the treatment of AFB. Although there is anecdotal evidence that indicates that OTC is being used for the treatment of AFB. So why is it illegal? Let's discuss the disadvantages associated with the use of the antibiotic treatments for AFB. The first one, when you apply the oxytetracycline to the beehives securely mixed with the sugar or other substances, initially for the couple of months, there will be no sign of disease, but the disease will start to return back between 3 to 14 months after the treatment. Few years back, Japan imported Australian honeys, but they all sent back due to the oxytetracycline residues in the honey. So it is very important to have a pure honey without any residues of antibiotics in it. And lastly, Penibacillus larvae has started to develop the resistance to oxytetracycline. To determine the state of the sensitivity in 2019, beekeeping industry requested us at EMAI to test oxytetracycline sensitivity of Penibacillus larvae in New South Wales. In 2005, the authors Wu et al. Published a research study, the Journal of Apicultural Research, where they have tested total 44 Penibacillus larvae isolates, and their study demonstrated that the Australian Penibacillus larvae isolates were sensitive to oxytetracycline. So we used the same techniques and the methodology in 2019 to find out whether the New South Wales isolates has developed any resistance against the oxytetracycline. The 2005 study indicated that the MIC minimum inhibitory concentrations to oxytetracycline ranged from 0.04 microgram per mil to zero point 0.6 microgram per mil. To determine the antibiotic sensitivity in 2019, we decided to replicate the methods which they have used in 2005 study. We began to culture 2019 NSW diagnostic samples. They were either honey samples or the larval bee smears, and with these we instigated our project. We also use the same agar dilution method what they have used in their study in 2005. To find out the minimum inhibitory concentration of the Penibacillus lavi isolates to oxytetracycline, we have prepared a few different concentrations, including no OTC, where there is absolutely no oxytetracycline, then 0.04 microgram per mil of the concentration of oxytetracycline, then 0.1, 1 microgram per mil, 2 microgram per mil, and 4 microgram per mil. In our study, we have tested total 47 Bacillus larvae isolates. In this picture over here, we can see we have used three different Penibacillus larvae isolates, which grew very well on the control plate, as well as on 0.4 microgram per mil, and as well as also on 0.1 microgram per mil, but did not grow on 1 microgram per mil. And therefore we can say the MIC minimum inhibitory concentration of these three isolates were 1 microgram per mil. Out of our 47 isolates, 35 did not grow on 0.04 microgram per mil. Therefore, we can say the minimum inhibitory concentration for those 35 isolates was 0.04 microgram per mil. In rest 12 isolates, the one isolate did not grow on 0.1 microgram per mil. Out of remaining 11 isolates, 8 did not grow on 1 microgram per mil, and last 3 stopped at 2 microgram per mil. In 2007, a research paper in veterinary microbiology reported that the Penibacillus larvae isolates can be considered to be susceptible to oxytetracycline when their minimum inhibitory concentrations are less than 4 microgram per mil, intermediate for MICs of 4 to 8 microgram per mil, and resistance when it is greater than 8 microgram per mil. And therefore, in our study, whatever we saw, we can say that all NSW isolate tested can be considered sensitive to oxytetracycline. Let's move to the second topic. As I mentioned before, there is some anecdotal evidence that indicates OTC is illegally used for the treatment of AFB in mainland Australia. So for the regulatory purpose, we, the laboratory at Elizabeth MacArthur Agricultural Institute, have introduced a new test to detect oxytetracycline in powders collected from beehives where the majority of AFB positive. This is a test kit what we have used to detect the presence of or the absence of oxytetracycline from the powder samples from the beehives. This is the test kit is normally used for the milk samples where they want to check the presence or the absence of the beta-lactamase and tetracycline from the milk. We modified this test kit to use for the powder samples collected from the beehives. This test kit is just like the normal dipstick method test kit or similar to the pregnancy test kit. The first picture is the instruction and the second picture on the right is what the test strip looks like. If the red line is visible, the sample is negative for beta-lactamase or the tetracycline. And when the red line is not visible, the tetracycline is present in the sample. Let's have a look at the results for the last 3 years. In 2019, we have tested a total 14 powder samples for the oxytetracycline and we found 11 positive and 3 negative samples. In 2020, out of 9 samples, we got 6 positive and 3 negative samples. This year so far, we have tested only 1 sample for the oxytetracycline and which turned as a negative. Based on these results and our study, it indicates that OTC is being used for the treatment of AFB in New South Wales. Let's move to our third research project. Outbreaks of AFB occurs frequently, but source of infection is often difficult to determine or in many cases may even go undiagnosed. So our aim was to develop a technique to trace AFB infections and its outbreaks. Now, we are all aware of the technology used to trace and control COVID-19 infections and outbreaks. In comparison to the other countries, Australia has been playing a very successful role in identifying the COVID-19 outbreaks throughout 2020 and 2021, which has prevented the high mortality rate seen in other countries. One important reason for this success is the ability to determine where the infected individuals acquired the virus. The technique used to trace COVID-19 infection is called genomic sequencing. This involves the analysis of DNA sequences, that is, the base molecules that make up the DNA of the bacteria or the organisms of interest. This genomic sequences technique is also used in tracing source of the food poisoning and the hospital diseases outbreaks. This genomic sequences would be also have an application in tracing our AFB sources. Let's discuss about the advantages of this GS technology. Comparison of genomic sequencing data from the AFB outbreaks would provide information to the link positive detections and identify strain clusters. These links and clusters could then be used to support the traceback and trace forward the activities by biosecurity compliance officers. When applied to the larger population, this can expose the transmission pathways and give biosecurity compliance officers powerful information to help to control the disease and its outbreaks. Additionally, an accessible online database where GS data could be uploaded and analyzed from multiple states, jurisdictions would allow AFP outbreaks to be more easily traced across the state borders. In this database, we have included the information relating to the B samples, including submission number, that is our unique identity code for our laboratory. Then we have added the total samples received, means like we received like 3 combs, 4 smears, 5 honey samples, like that. Then type of the sample. So our positive Bacillus larvae, either we picked from the smears or we picked from the honey or from the comb, and then sample number, then submitter's name, owner's details, property address, hive location, suburb, postcode, etc. EMAI is the diagnostic center for the bee disease diagnostics in New South Wales, including AFB diagnostics. We hold an extensive range of the Penibacillus larvae isolates. This includes isolates from the most Australian states and more recently isolates detected in Northern Territory. We have also got a collection of the freeze-dried isolates from 1982 to 1998. The main source of Penibacillus larvae isolates for this project were larval bee smears, brood combs, interstate honey samples, and roughly 200 historical freeze-dried isolates. The successful outcome of this project, we can identify more cases and the sources, and it can help to reduce the number of the infections and prevent the future outbreaks. Validated genomic sequencing tools and online databases will help link cases together across the Australia. Also gives us an ability to identify even minor variation between the species. In the beginning of 2020, New South Wales Department of Primary Industries at EMAI commenced this project with the collaboration with University of Technology Sydney. This project led by research scientist Dr. Daniel Bojema, bacteriologist Dr. Anya Deutscher, and the bee experts Dr. Michael Hornitsky at EMAI. Funding was obtained through the Australian Centre for Genomics Epidemiological Microbiology, a research centre with the funding with UTS and New South Wales DPI. Further funding is being sought to straighten this work and construct an online database to identify the linked AFB detection and aid AFB outbreak tracing. I would like to acknowledge our bacteriology team at Elizabeth MacArthur Agricultural Institute and Department of Primary Industries for all their support and the time. Thank you.